Hello, my geniuses. Welcome to another biochemical lecture series. Today is your great science tutor Lorenzo, and today we are going into our first topic in molecular biology, which is molecular probe. So we'll be doing molecular probe today. So this molecular probe is the first topic we'll be doing under the molecular biology playlist. So if you are new to my channel, make sure to subscribe and turn on your post notification button so anytime I drop any video, you'll be sure to be notified. If you want me to make a video for you, drop it down in the comment section. I promise I'll make that video for you. And if you want to ask me any question, or you want to ask me any personal question, or you have issues understanding, just send it to me privately on my WhatsApp number, which is on my channel. So I promise I'll get to you. You get it now? So let us jump right into the video. So first of all, what is a question? What is a molecular probe? If they ask you what is a molecular probe, now this involves the use of a single-stranded DNA and RNA fragment. So listen, I'm taking a strand of a DNA. It could be a strand, just a template or a coding strand. I'm taking a strand of a DNA, and when I take a strand of the DNA, are you getting me now? I want to inject it, are you getting me now, to another part of a DNA in order to dictate the presence of a complement or nucleotide sequence. Are you getting me now? So you are taking a strand of a DNA and an RNA. It could be an RNA structure, just one strand. Because you know we have two strands. We have the template strand, we have the coding strand. You get it now? Because a DNA molecule is a double helix structure, and we have two different helix, two different strands complementing with each other. Should be, you know that, using what we call Chango. So when you want to dictate a complement base, so maybe I take a different DNA strand, maybe I have a different DNA strand like ATGC, or just a random DNA strand, and I want to dictate the complementary sector or the complementary factor to that strand. What do? I take a probe. A probe we have, I will take the probe and make sure the probe is complementary. So if I take the probe and I insert it into the normal or the target DNA sequence, I want to check for complementary. So I am checking for complementary by what, taking my probe. My probe is my sample DNA. Are you getting me? My sample DNA I am using to inject into the new DNA. Okay, now so if I take it inside, I might run it through various DNA systems. So when I run it through the various DNA system, I want to check for the one it matches. So it's just using a sample DNA to check for which part of the whole strand in a particular section that it rhymes with. You get it now? You get it now? So that's what I'm trying to do. Taking one strand of a DNA and checking for where it can complement itself in the whole strand. So if I have a sequence, a long sequence with about 100 nucleotide bases, if I have 100 nucleotide bases and I want to check for which strand, and me, I have a strand of about 10 sequence, and I want to check inside that 1 to 100, where will the 10 or where will the 5 be complementary? So if I get to the part it is complementary, that sample DNA I'm using to go is what we call a molecular probe. So a molecular probe is used to check for a complementary nucleotide sequence using a what? A probe. So the probe is the sample DNA or RNA I'm trying to inject into the system to get a complement. So now it works by what we call hybridization between the complement DNA and the RNA sequence. So it works by hybridization. So it's just helping you to check which part of the DNA or RNA sequence that is complementary. You get it now? So what are the components here in a molecular probe? You have a known nucleotide sequence. The one you have, the sample DNA or RNA sequence, is called the what? The probe, the nucleotide probe you have. And you get the label, which is the one you are trying to do, is what we call a radioisotope or a fluorescent dye. I get it now. So one of all, you have to have a known nucleotide sequence and also the label. So it's one. Either you have a probe, so you have a probe and you have a label. A probe and a label. That's a complement of a molecular probe. So you have to have a probe, which is a known nucleotide sequence, and you have a label, which should be a radioisotope or a fluorescent dye. So what is the principle of operation? How does it work? A molecular probe binds to a what? A complementary what? Nucleotide sequence to what? The process of what? Hybridization. I get him now. So the work of it is that when you inject it into the system, what happens? The probe will bind to a target sequence, and when it binds to a target sequence, the label will now what? Enable the detection of a nucleotide. So what are you doing now? So for you to have a successful molecular probe, you have to have a probe. the probe itself, which you want to use to send into the system to detect the complementary, and you now have to have a label. A label is that when the probe sees the targeted DNA sequence, what will happen? It will now use the label to now label this part, that this part is a nucleic acid what sequence. You get it now? So do you understand? It's very, very simple. You see the probe in the picture. This is the probe. The one that is the labeled single-stranded DNA or RNA. It's just a single strand you made. Then now what happens? You now inject it into the target DNA or RNA to check where it will be complementary to. So when it sees where it is complementary to, so it will now bind to it and become complementary by what? Hybridization. So when it binds by hybridization, what will I do now? The radioisotope or the fluorescent dye will now highlight that place. If you highlight that place, now you will now label your results that this part in this particular DNA sequence is what? Complementary. Are you getting me now? It's very, very important. So what is the application of molecular probe? One, you use it for what? Genetic research and gene mapping. Maybe I'm carrying out a genetic research, and in the genetic research, I'm trying to what? Map a DNA. Maybe somebody is having maybe Down syndrome or any particular genetic abnormalities, and I'm trying to map out the DNA. So what do? I make a probe in the laboratory. Are you getting me now? After I make a probe in the laboratory, I map out the patient journey with a particular genome device. So when I want to do it, I insert my molecular probe inside, so that my molecular probe will go to what? Bind to that specific fragment, so that to detect which part of the patient's chromosomes or which part of the DNA sequence is what being affected. So it's used to just highlight, it's used to know a particular strand of DNA that is affected in any chromosomal abnormality. So you can also use it for research to detect. And when you use it to detect, you can also use it to treat. So it's also used in biotechnology to reproduce using biological organisms to produce so many things that we use in medical, agricultural, and industrial environment. You get it now? So number two, what is it used for? It's used for cancer diagnosis. Like I told you, it's used to just map out the sequence in which there's highly abnormal or proliferation. So when there's abnormal proliferation of cells which causes cancer, what happens? To mark out the part that is what? Being that the part where the cancer starts growing, you can use a molecular probe. So a molecular probe is just a detection device. Finish. It's just a detection device because you make a sample of a DNA or RNA fragment inside the laboratory and you inject it into the patient or the individual genome to what? Search out the place that where you have the abnormalities or where the origin of the abnormality. You get it now? So it's used in cancer research. It's used in genetic diagnosis and mutation. Like I've always said, it's used to get out the part that is damaged, the gene that is damaged doing what? Replication or transcription or translation. So it's very, very important. Also, when there's genetic mutation, when there is any destruction of any gene doing what? Replication or cell division or cell cycle. So you use the molecular probe to detect. So it's just a detection device. Also in DNA fingerprint, Shebi, if DNA fingerprinting, what happens? So when everybody has their own personal DNA, everybody have their own personal fingerprint. So when you, especially in the for investigation, maybe an individual left his print in doing a murder case, what do? They take the print, run it through DNA sequence, and when they use a DNA probe, they send it, use that print, put it in the machine, and now use the DNA probe to check which of the individuals in the file, in the country's file, has done the particular crime. They use the DNA probe to what? Send it to various people in the distance. So they take the prints. The print now they take from the person is a probe. Are you getting that? When they take the print from the probe, they send it to many people DNA sequence. Anybody it matches to, that's the person that committed the crime. You get it now? So they use the individual or the killer's print. They use the individual killer's print to make a probe. When they use it to make a probe, they send it to various database of DNA that they have gotten from everybody in the country, like in maybe in countries that are developed like America. So this is used to detect the killer. Also is used in detection or an identification of infectious disease, the same thing. Anytime diseases enter into the body, what happens? They go to bind or they go to hide inside a particular part of the body and start doing unnecessary replication and transcription and translation. They start doing so many genetic movement in the body. So also we want to know where the We want to know where the bacteria or the virus or the fungi started his proliferation and want to use it to highlight that place so that we can get a drug that can use to kill it. Okay, now, so we have advantages of molecular probe. Why? Now, the next one is the advantages of the molecular probe. One, it's highly specific to a particular DNA and RNA sequence. We have said it. So you take a particular DNA and RNA, send it into the system, it attaches and get a specific fragment where it is what complementary by hybridization. It's used in repeated detection of genetic material. We have said it. So it's very, very easy. That one is they are used to detect. Three, it can detect very small amount of what? Of nucleic acid sequence. So if you want to check in any, maybe it could be a crop, it could be antibiotic, it could be anything, you can take it through the body. And that is why anytime you take a drug is specific. Okay, if this drug especially is used like ergot, let's say a particular ergot which is used, some ergots are used for headache, some ergots are used for the womb, you get it now, for the uterus. So how do scientists know that, okay, if I give this drug, it's going to do this thing? Why is it that if you take a drug, it can help you to stop headache? Why will it go to the brain? How did he know it's supposed to do this kind of thing? That is how you use a molecular probe for it. It's used to also in early diagnosis of diseases. It could be infectious, it could be cancerous. It's widely applicable in research and forensic science and clinical diagnosis. So now, what is the limitation? It's expensive to develop. Are you getting now? It's expensive to develop and use. So for you to acquire any genetic machine or to even make a molecular probe by yourself, the company or the individual has to be rich, because the material to synthesize a probe or the material used to do in genome mapping or in genetic research is very, very expensive. That is why the WHO and other private organizations help to finance all this research. Now, it requires expensive laboratory equipment. We have said it. Yeah, so one of the genome machine that we use to do all these things. So three is that it requires skilled personnel for accurate interpretation. Yes, so you have to meet a genetic scientist or a molecular biologist or somebody that is specific into the research. It might not be a doctor or a nurse or any of the medical professional. It will be somebody that has trained and studied to know and to interpret genetic information. The risk of contamination lead to false results. So remember, you are trying to get a DNA sequence, and that DNA sequence should be highly purified. If not, when you insert it into the body, it could malfunction and give you a bad result. Radioactive probe may poses safety risk. Yeah, because it will pose safety risk in the sense of, remember, when your body is exposed to radioactive substance, what happens? It can cause another genetic mutation in another part. So let us say you are sending in the molecular probe into the body. If you send it into the body, what will happen? It could cause another thing by causing another mutation in another organ. Trying to, even though it works, but it can cause other mutation, because when you expose the body to radioactive isotope, no matter how the radioactive isotope is so minute, it could also cause some problem. You get it now? Also, it reduces sensitivity if the targeted sequence is mutated. So like I said, the sensitivity only works when there's no mutation, but it is used to detect mutation. But if you want to use it, because you can use some more probe for treatments too, I get it, also in gene therapy. So you only use it to detect, and but when the sequence is mutated, it can give you a false result. It is kind. Now the last one is time-consuming sample. The last one, time-consuming sample preparation may be required, because to make a molecular probe, it takes time. It takes time to make any molecular probe, and it's sometimes days, weeks, months to make one probe, because you have to be accurate. You have to ensure quality control before you can make one probe, send it into the body to even dictate what it wants. You get it now? And now, hybridization. How probe binds to target. Remember, I told you, you take a probe, put it to the target DNA, it binds by hybridization. You dictate using what? Fluorescent dye or radioactive isotope, and you record your result. And now, by hybridization means that it must be complementary. If it is not complementary, like you see here, A must bind to. So if you have a probe which is ATGCCA, this is a probe now, and you are sending it into a DNA sequence, it must bind to TACGGT. You get it now? That is how it is. So the binding is by what? Hybridization. So now, in conclusion, now what will we say? Molecular probe are labeled what? Single-strand DNA or RNA fragments. So they are single-stranded DNA RNA fragments, which you send into the body for what? Complementary. You have to make sure that it is complementary, because anytime, remember, for replication, there must be formation of a bubble, which means the two DNA strand has to open. So if the two DNA strand are open, what will the probe do? The probe will go to bind and check where it is what much more complementary. Are you getting now? It is a very important tool used for research, disease diagnosis, cancer detection, DNA fingerprinting, and identification of infectious diseases. Despite the advantages, what happened? They have limitation. Cost is a limitation, which we talk about. You have to be highly funded before you can do this. Also, technical complexity, which means the individual which will be interpreting these results, the environment needed to interpret these results has to be what top notch, and also a need for what specialized equipment and training what professionals. So I hope it all made sense to you and you have understood everything I've talked about in what molecular probe. It's very, very simple. I use a probe, put it inside a DNA sequence and check where it is complementary, and use my label, which is the fluorescent dye and the radioactive isotope, to what? Determine the spot that is complementary. Finish. Yeah. So if you are new to my channel, make sure to subscribe and turn on your post notification button so that anytime I drop any video, you're sure to be notified. If you want me to make a video for you, drop it down in the comment section. I promise I'll make that video for you. So we'll be going to the next one, which is gene therapy. Stay to my video, stay to the last, because I'll be giving you useful tips. So this is the first topic in molecular biology. So we'll be going to gene therapy. From gene therapy, we'll do biotechnology. From biotechnology until we do various topics. And you learn a lot, because this is the future of what? Medicine. Genetic research. It is the future of medicine, which we want many people to go into, because there are new diseases that are being evolved. Are you getting me? With genetic experience, you could do a lot with it. So I'll see you in my next video. Bye bye.